Hello students, we are coming to the last part of the chapter, that is obtaining the foreign gene product commercially. So first of all, what we have just made a recombinant DNA. Then this recombinant DNA was inserted inside the host, one heterologous host. Heterologous host means those hosts where the products are actually produced. They are of not their own, they are produced actually due to the insertion of a foreign gene. With the help of this vector, they have been inserted. Then after that, that host will produce the products. That products will be completely of that gene that was inserted. So after that, whenever it was inserted, they will start to grow, they will multiply. So for all these processes, what we need a culture, a continuous culture plate, so that our nutrients can be provided, so that the host can grow, host can multiply, and host can produce the products, our desired gene product. So for that reason, a lots of qualities should be present so that we can able to extract our desired product. So for that reason, we need the nutrients, we need the proper sterile environment, we need the temperature, pressure, pH balance. Then only it is possible that in large quantities we can able to extract the product. So for that, for the commercial purpose, we have one technique, one actually method within which in large quantities, suppose from 100 to 1000 kgs of products can be extracted by the use of bioreactors. So what are the bioreactors? See, bioreactors, the bioreactors are the large vessels where in large quantity products are produced by maintaining proper environment. Okay, so these are the bioreactors, where these are actually the large vessels where we will continuously supply the fresh nutrients. Then those nutrients that will be taken by the host body, then the host will grow properly in the controlled temperature, in controlled aeration, in controlled pH, so that the pH can be maintained. In all of this, they will be just multiplied. Then after that, in desired amount like 100 to thousands of kgs of products, we can able to extract it properly. So for that, we are going to have two types of bioreactors. First one I have drawn already here, it will take more time. So it is the simple stir tank bioreactor, and another is the Sparse stir tank bioreactor. So these bioreactors, the main basic principle is the same, that we are able to extract the products. How can we will able to extract the product? It is with the help of the large inflow of continuous nutrients. Okay, we have to continuously flow the nutrients, then only it is possible that we can able to extract our products. So for that reason, what we are going to have, this is the simple stir tank bioreactor, where you can see a shaft, and along with the shaft some blades are there, right? So these blades, whenever this motor will run, this blade will start to circulate. And here we are going to have some inlets, okay, with the help of what? Here some inlets are present. So with the help of the inlets, we will continuously have the nutrients into that tank. Large, it's not of a simple this much vessel, it is of a large tank, maybe of suppose 1 meter length, breadth, that area, or even more than that. In that large tank, we will have a shaft. This is the shaft, and along with the shaft some blades will be there that will start to move, that will start to rotate with the help of this one motor. Then along with that, there's going to have a thermometer. So that thermometer will check the temperature. If the temperature is increased, that suppose that that thermometer will indicate that we can cool down it, or if the temperature is already cooled down and it needs a optimum temperature, then this temperature can be once again maintained. So that's why a temperature maintaining thermometer will be present. Then after that it is going to have also one acid base control, so that we can maintain the proper pH. It should not be more than more acidic, it should not be more basic. So for that proper host, whatever the optimum pH is needed for that host to survive, that pH will be maintained. Next one, a steam for sterilization. Proper aeration will be there, proper a flow of steam will be there, so that it can purify if some foreign particles are growing. So for that we are going to have the steam for sterilization. Next one, we will have the culture broth, then where actually they will grow. Next one, flat bladed impeller. So these are the flat bladed impeller, that will actually nothing but the blade. Whenever it will rotate, then continuously, vigorously this nutrient will start to move, and it will equally distribute the nutrients to the host, so that it can grow. So this is one bioreactor where this shaft is there, and with this shaft will rotate with this running of the motor, and these blades start to move like this. The blade will start to rotate, and this way it will able to properly maintain environment, so that our desired gene product, product, maybe the gene product or maybe the protein, that can be extracted. Next one, here we have another technology, we have another type of bioreactor, that is the Sparse stir tank bioreactor. Here just a thing is different, that is that shaft. Here in this shaft the blades will be not attached. Here rather that it will have some bubble producing. That means suppose in the aquarium what we have a bubble that will be released, and this bubble will burst, release the gases. Similarly, this is the same type of technology. There will be have a sparger. So with the help of the sparger, the bubbles will start to release, and it will go burst, release that gas, oxygen, for the product to be formed. Otherwise, bubbles that will increase the surface area, transfer area, then this surface area is increased so that proper oxygen transfer can be there. Sparger will produce that bubble. Then after that, gas entrainment. So whenever they increase the surface area like this, then this gases that can be also trapped from the surface. So from these two bioreactors, most commonly used bioreactor in the biotechnological laboratories companies, that is the simple stir tank bioreactor. So with the help of the simple stir tank bioreactor, normally large amount of products that can be extracted. So how can we able to, suppose after that we have produced suppose millions or thousands of kgs of the product. So after that what we are going to have the downstream processing. So what is the downstream processing? It is actually a technique by which we will we will first of all take the product from the cells. So we will extract our product. Okay, we will extract it. Then after that we will purify. Okay, then after that we will completely see whether it is we will go for the safety measures also. That means we have to purify it. We have to first of all check the quality. Okay, quality and safety. Okay, so we will check all of this. Then after that we will able to extract the product for the commercial purpose. So in the bioreactor, suppose the cells, they have produced the protein. So first of all we have to do one thing, that extract the protein from the cell. We don't need the whole cell, but we just need suppose the protein. So first of all from the cell we will collect the protein. Those protein will be purified, right? Purified. After that we will check the quality of the protein, whether it is the normal, the desired protein or not. That thing will be checked. Then after that we will go for the safety measures also, that it will be tested on some another organism or it will be tested in laboratories. Then after that it will be packaged, then it will be sent to the commercial areas, so that the different markets, they can take the product and it can be available for the people. So this is the whole technology by which we can able to produce thousands of kgs of product, desired product, with the help of the bioreactors. So whatever we have think, this is the whole chapter. This is the biotechnology principle and process. So we have completed the chapter. I hope you understand, see more and more, study more and more, and you will be able to understand it. Try to recall, practice it 2-3 more times. Okay, study 3-4 times, because see, other chapters like strategies, microbes, they're not that big chapters, they're very small chapters. This is one chapter which will carry a little weight. So from the weightage from your subjective as well as objective examination, this will carry at least some more marks than that of this chapter, strategies and microbes. So that's why study hard, you will get more. So that's all about a chapter. I hope you understand. Thank you.